The release of extracellular traps by neutrophils and other immune cells is important in innate immunity, but also strongly linked with inflammatory pathologies. Very little is known about this process in macrophages, although these cells play a critical role in inflammatory response. This protocol provides a primary human in vitro model of macrophages extracellular trap or mass release. It provides a model for us to study a potential physiological role of this structure in vivo. The main advantage of this technique is that the cells from this protocol are primary cells, isolated from human buffer coat preparations. There is no priming step required to differentiate the monocyte into macrophages, which is contrast with other monocyte cell line, such as THP1 cell line. This protocol is easily adapted to other immune cells, including isolated neutrophil and microglia. Extracellular traps produced by macrophages are very fragile, so care must be taken between steps to preserve the integrity of the stained traps.
Under sterile conditions, prepare M1 priming medium by adding to the complete RPMI 1640 culture media interferon gamma to the concentration of 20 nanograms per milliliter and lipopolysaccharide to the concentration of 1 microgram per milliliter. Prepare M2 priming media by adding interleukin-4 to the complete RPMI 1640.